Thank you, Naomi. Here's my presenter disclosure, which is a little bit long. I'm not going to read it, but it says that I work a lot with companies and I don't get much money from them. So this is the flyer that we got, many of us, at this meeting. And when I looked at it and all the words, it's a word cloud kind of flyer, I noticed that there was nothing to do with taste and flavor. And yet this is the most important thing for a nutritionist to understand. That's our argument today. And I think it's so important because no food, no matter how nutritional it might be, is going to be consumed unless the flavor is preferred. So I'm going to give I'm going to go quickly over a lot of biology of taste. Basically, my purpose here is to give some feel for the breadth of the field, and so I'm going to go through things fairly quickly. First, I want to talk about the word taste. It comes in two kinds of flavors. One is the taste as a sensory system, which consists, as Naomi said, of sweet, salty, sour, bitter, umami, and maybe a few others. And then there's taste as the flavor area, and that would include olfaction as well as chemesthesis or chemical irritation. So the flavor senses, I would argue, are the most critical senses of all for controlling food acceptance. And during human evolution, the most important decision anybody ever made every day was whether to put something in their mouth and swallow it or to reject it. This was a life and death question. And obtaining enough food was the primary goal, maybe one of the two most important goals, one to get enough food, the other to reproduce. In many societies, obviously, this is no longer the case. The need to get enough food has been supplanted by the concern about getting too much. But it's important to remember that there is still large parts of the world where it is still an issue of getting enough. So the reason that I think that it's important to look at the excess consumption is because of the role that flavor-driven diet choice has in a wide variety of diseases, of which you're all familiar with, and I won't read them. But these are the concerns that we have, and the concerns that are sort of the crux of the matter being the difference between our evolution and what we were driven to consume, and the availability of things we have now. So this is a picture of the flavor senses. I don't know whether anybody can see the pointer, but I'll give it a shot. Doesn't seem to work even. There it is. So taste, as I'm going to define it, is the conscious perceptual taste is mediated by receptors in the tongue and the palate. Smell is mediated by receptors way up here in the top of the nose, and the odors can come through two ways, once by sniffing, the other via the retronasal pathway, which is mostly what we have for food. And then there's chemesthesis or irritants, which are throughout the whole body, but are particularly on mucous membranes. So what are the functions of taste? Well, there's one, and this is a dedicated sense. There'll be an interesting talk on taste in the rest of the body, a whole session on that. I think it's tomorrow. But at least for the conscious part, I think the taste has a single function, which is food choice and utilization. For these irritation senses, there's also, it seems to have a major role in warning us that something might be dangerous. But for humans, at least, food choice and utilization is important because many people obviously like the burn of hot peppers. And one interesting fact that, at least I think it's a fact, is that of all species, we are the only ones that seem to like to hurt ourselves with pain. And we can try to figure that out. I don't know the reason. If we look at olfactory functions, there are many more. So this olfaction is a much broader sense, one that functions not only about food, but also about mate choice and all of these other variables. Oops, I went too far. So what about the taste quality descriptors? and these are the same ones Naomi put out. I'll have a little bit more to say about them in a moment, but these are the consensus qualities for the sense of taste. For chemesthesis, it's a little mushier in terms of what these exactly are, but these are the sort of the general ones that we think of as stimulating what we call irritation, but can be cooling, burning, pain, tingle, numbing, itch, and chemical touch. But look at the odor quality descriptors. Here's just a small list of all the quality descriptors, and one of the interesting things about smell is we don't have words for smells. 90% of these things, which you can't read at all, are objects. It smells like rather than having words for the smell, and that's an interesting difference between taste and irritation. So the dimensions that we respond to for flavor are listed here: sensitivity, intensity, location, timing. Some of this will be discussed by the next speaker, John Hayes. I'm going to concentrate really on quality and the hedonic or pleasurable aspects of the sense of taste. So first of all, though, I want to give a brief overview of olfaction and chemesthesis, just so we have a little bit of background on that. And on the cover of your program, this set of pictures were produced by Monell. So here's smell. These are the receptor cilia in the top of your nose. They're bathed in mucus, and on the end of these cilia are molecular receptors. And these molecular receptors were identified first in 1991 by Buck and Axel, for which they won the Nobel Prize in 2004. And the most remarkable thing about this is that in humans, at least, there's 300 to 400 separate receptors, each depending upon a single gene, so that around 1% of your genome is devoted to the sense of smell. The smell space is vast. There's many, many things that stimulate smells. And one of the really remarkable variations within biology is the number of olfactory receptors in different species. So this shows the elephant has the most olfactory receptors. Down at the bottom, we have dolphins that have none. And humans are somewhere in between. But this is a huge huge numbers of receptors, which is very different than the other senses. The way olfaction works, we really don't totally understand, but there's at least presumably some kind of combinatorial code where each receptor can detect many different odorants, and in the glomeruli, which is these. So these are the receptors up here. They're colored based, each color represents a single receptor, so we're only talking about 10 here rather than 400. And each particular receptor, each color, is connected to only one of these round circles here in the olfactory bulb called glomeruli. And so there's a direct pathway there, but each receptor may stimulate, may be responding to different odorants, and each odorant is detected by different receptors. So it's a very complex system. So we can just see some sort of cartoon here of response to rose, and you can see that the rose odor stimulates only a subset of these receptors. This is an absolute cartoon, very, very complicated system. One thing that I did want to point out, though, is even though we're in principle able to detect millions, billions, it's been suggested, of odors, you can't really detect individual odors in mixtures. And this is a famous set of studies by David Lang and his colleagues, suggesting that if you take, say, 4 or 5 different smells and you mix them together, nobody can pick out what the odorants were that are in that group. And so we really don't understand how these individual odorants come together to form even different percepts. This is a system that is a long way from being understood. Now let's look at chemical irritation. It's really a somatosensory system. It goes along with touch, proprioception, vibration, pain, hot and cold. So it turns out that these are transduced by different kinds of receptors, but the chemicals have the ability to cause all of them to be activated. And so each of the three sensory modalities is chemically sensitive, and therefore chemicals can stimulate essentially touch and pain, and this is a very, very important component of food, and one we actually often ignore. One of the major receptor classes are the TRP receptors, and TRPV1 and TRPA, one of the most famous ones. And they were first discovered as temperature receptors in the body, and then it turned out that they also respond to chemicals. And so when you taste hot peppers in your mouth, it burns, and it is exactly the same as if you were putting a little bit of fire into your mouth. It's the same receptors that are doing that. So there's a whole class of these receptors, and these are of great interest for a variety of reasons, but I think they're really important in food choice and ones that we don't attend to sufficiently. Now I want to move on to the main area that I wanted to discuss, which is taste. And this is one of my favorite quotes, because I think this is the answer to, or this is the problem at least. So, taste qualities and basic tastes. As Naomi mentioned, these are the consensus ones. They're based on what I have a consensus criteria for being basic tastes, if one looks back in history, one finds that this set of basic tastes has a very, very long history. This is a list that Aristotle put out. The top four are the sweet, sour, salty, bitter. Then those below it, excuse me, interestingly, are the kinesthetic qualities. And Aristotle and many others early on mixed up chemesthesis, that's the irritating property, with taste. And it makes sense because these are things that stimulate the mouth and didn't know much anatomy, so why would they think that they were different? Galen had the same general view, and Galen probably was influenced by Aristotle, but here if we look at India, thousands of years ago, exactly the same set of basic tastes, the sweet, sour, salty, bitter, along with a couple of the irritants. We can look at a couple of more other societies here in Borneo. Again, the same list. This one actually seems to have maybe a notation of something that's like umami. And so many of the psychophysical studies are consistent also with this historical study to suggest that these basic tastes are perceptually separate, perceptually independent, and form something that has been found throughout history as basic. The other consensus criteria have to do with functional significance. What are they there for? And much more recently, molecular and neural specificity. And the consensus for basic tastes, and this is, certainly all of you are familiar with this, is sweet taste, presumably evolved to detect sugars and calories, bitter taste to detect poisons or dangerous substances, salty taste to detect sodium, sour, I never quite know what sour is there for, but maybe unripe fruit and spoiled food, and umami, supposedly amino acids and protein. And these are very, very important nutrients, and ones that seem to show across species that for animals, particularly that eat plants, these are a list that we find consistently. Now, there are other candidates for basic tastes, and they named several times, and we are somehow going to have a debate somewhere in some other meeting as to whether these would be considered candidates for basic taste. I think no. I know at least one person in the audience, Rick Mattis, thinks yes. But it's an interesting debate. But my argument is that these may all interact with taste receptors. They obviously respond to important nutrients, but they lack criterion number one, which is a chemical perceptual saliency, which I think is the most significant criterion for the basic tastes. So let's talk a little bit about sweet taste. was beginning in about 1999 and then 2001 that we finally identified the sweet taste receptors. And this is a cartoon of the receptors. Basically, on the left is a sweet taste receptor. And it's made up of two receptor molecules that form a pair. And they can respond mainly in this binding pocket here. So this is, if you think of the tongue, this is the outside. This is where the saliva is. Inside is inside the cell. These molecules can interact with at least one of these two different taste receptor proteins. And one of the interesting things is that this doesn't show it so clearly, but one reason we're able to taste so many things, or so many things are sweet, is that these receptors can actually interact with other parts of the molecule. For example, cyclamate doesn't hit this binding pocket, it interacts down here. This is, over here is the umami or amino acid receptor, and you can see that they share two common protein receptors, and they have one different one. This is T1R2, this is T1R3, this is T1R3, and T1R1. And the binding site for T1R1 for the umami, for glutamate, and the compounds that enhance it is this unique T1R1. So I'm going to get rid of those compounds that are confusing and just talk about these receptors. And I want to talk a little bit about comparative aspects of sweet taste, because one of the issues is that we talk about sweet as being something that detecting calories and makes a lot of sense, but what's the evidence for that? So this is an animal that loves sweet taste, and you can obviously see he's smiling. I think it's pretty safe to say that most species, and this can go all the way down to bacteria that respond to plant materials, have something resembling a sweet taste. So this is a creature that I studied many years ago, and it turned out that it doesn't have a sweet taste. We could find no evidence that they liked sweet taste, and then my colleague Joe Brand and Shali demonstrated about 10 years ago now that if you look at the taste receptor, one of those two taste receptors, the T1R2, which is the one that was unique to sweet taste, here's the dog taste receptor, basically showing these components of the receptor all working quite nicely. However, for the cat, there's been mutations that result in this receptor no longer functioning. And this is the explanation for why cat food manufacturers don't put sugar into cat food. They can't taste it, and in fact, it will make them sick. So basically, cats have lost sweet taste function because they've lost the function of this particular receptor. And this is a real Garfield cartoon demonstrating that. More remarkable to me is the work by Pei-Hua Zhang and his colleagues demonstrating that this has happened multiple times in species that eat only meat. So this is a list of various species that eat meat, and what's happened is that they've independently developed these mutations. So it appears as if when an animal moves from a omnivorous diet to a pure carnivorous diet, which obviously happened millions of years ago, there's no longer selection to maintain the sweet receptor as operable, and it is lost. And it's lost independently. Each species did it separately because the breaks, where the red are on that slide, are in different places on the receptor. There's also, even within species, large differences in response to sweeteners, and particularly for non-nutritive sweeteners, those seem to be really aimed at the ones that we use, aimed at the primates of the old world. So here's some new world primates, and none of these taste aspartame as sweet. Their receptor is built in such a way that it somehow doesn't detect it. However, if we look at old world primates, all of these detect aspartame as sweet, I think. I haven't one of them I haven't tested. Finally, showing the power of sweetness is the remarkable issue of birds and sweet taste. So this is a hummingbird. Obviously, we know it likes sweet, but it turns out that all birds, during the separation between birds and reptiles, lost that T1R2 sweet receptor. So how is it then that the hummingbird is able to detect sweetness? And it turns out, and it turns out that what the hummingbird did is it has somehow repurposed the umami receptor into a sweet receptor. Now, this may be true in lots of species of birds, we don't know, but again, it shows that the fact that as this particular class of birds got into a niche where they were able to exploit a different source of calories, they developed, they somehow evolved to detect sweet by taking the amino acid receptor, and presumably the data from the paper show that they no longer detect amino acids or umami. Now I'm going to move to salty taste, and salt is a fascinating ethnological issue. It's been argued that it's the primordial narcotic. It's obviously is a huge concern about excess salt consumption, and particular worry now that when it's no longer hard to get, there's too much of it, and it provides a danger. And these are some cross-cultural data from the very famous Intersalt study, showing that in some cultures where salt intake is very low, so is hypertension. But in the majority of countries where it's around about the same, there's a great range in hypertension. And this can be interpreted in many ways, and it's been argued as to whether this shows that salt is dangerous or is not. But this is the proof of it. Salt is the real killer. So this struck me as just as I was first thinking about this meeting. Here's two articles from the New York Times within 5 days of each other from two of the very prominent nutrition writers there, Gina Kolata and Jane Brody. And I'm not going to tell you to read it. I can just tell you that they come to remarkably opposite conclusions about the importance of salt for dealing with hypertension and blood pressure. So if we look at chemicals that are primarily sweet, and I kind of mentioned this, that many things are primarily sweet, and you can contrast that with salt. Very, very few things are primarily salty. Sodium chloride, lithium chloride is a great salt substitute, but it's poison, so I wouldn't use it. KCl is an interesting one, and I'll come back to that in a moment. So what are the receptors for salt? Well, this has been suspected for a long time to be a channel. This is the same channel that's involved in salt detection in your kidney. It's called epithelial sodium channel. And in 2009, two different groups clearly demonstrated that this is involved in salt taste. However, this is not the full answer. There are other issues that are confusing here, and it's thought that maybe different levels of salt activate different receptors. There's issues about what sodium deficiency does to these channels, and there's the fundamental question that we don't understand. Is this ENaC is responsive very specifically to sodium, and if that's the case, why in the world or how in the world does KCl, potassium chloride, taste salty? We don't know. Salt, though, isn't a magic ingredient, and it does much more than give saltiness to a food. And this will come up in other talks as well quite a bit. This is a famous study by Marianne Gillette showing that if you measure a variety of perceptual characteristics of a soup, again, the aroma, the fullness, thickness, saltiness, so on and so forth, here's the soup, and here's what happens when you add salt. So you may make it saltier, of course, but you also add thickness and fullness, you reduce metallic and bitterness, and add something called balance. But it's providing much more than just saltiness. And this, as the other speakers will more fully discuss, is really a fundamental issue in terms of how you reduce salt in foods. One of the things it did, I showed you in the last thing, is it reduces bitterness. And this is a study we did a number of years ago showing if you take a sweet stimulus, and this is how sweet it is. This is just arbitrary numbers. And then another compound, which is bitter, urea, and this is how it's bitter and not sweet at all. You mix the two together, and in this particular situation, probably this mixture suppresses, so you have less sweet and less bitter, and this is probably going on centrally in your brain. But it turns out if you add salt, and this is not even a salty salt, it's just sodium of in any way, you suppress the bitterness, and that's at the receptor level, and this enhances the sweetness. And so you can, and this is the simplest of all foods, but if you think of this as adding salt to make food sweeter, and this is the problem. You can't just take salt out because you're going to reduce and change all sorts of other factors in the food. A little bit about salty comparative salt taste. It appears that animals that eat only meat, or mostly meat, the dog is not a strict carnivore, but mostly meat, can recognize salt, but they have no response to sodium deficiency. And that's thought to be maybe because they never become deficient, they starve first. And so there's, again, no pressure for them to recognize salt as a particularly tasty thing. I'm particularly interested in, and hopefully one of my colleagues is going to be studying this, is to what amount of salt are consumed by our closest relatives. We do know that if you feed chimpanzees high salt diets, they develop elevated blood pressure. But we don't have any idea of how much they consume in the real world and how much they would choose to consume. And this is, I think, an interesting question that needs to be pursued. Now, a word or two about umami taste. This is the most recent addition that people seem to generally believe is the case. But even here with umami taste, it's a more subtle perceptual difference. This is the quote from the Akita, who was the first to identify glutamate as the umami taste. But here, too, we have really remarkable species differences. And this is, again, a profound question in my mind. For humans, the umami taste is aimed almost primarily at the amino acid glutamate. But for other species that have been tested, and there are not that many, I have just two here, cats and mice, it is a much more general amino acid receptor, responds to many different amino acids, which makes much more sense based on the argument that it's to detect protein. Why is the human one so specific? What's special about glutamate? And a clue to that, perhaps, is that glutamate is very high in human milk, and there's some relationship between that. But this is really speculation, and we don't know too much about it. One other thing I would say, though, about MSG, an important component about that, and again, making the concept of taste much more complex, is that perceptually, it is not only gives a taste, but it also gives a feel. And these are quotes from a very old set of studies in the US from the 40s and 50s. But all of these people acknowledge this, that there's a feel to glutamate, not just a taste. And some original studies done by Yamaguchi at Ajinomoto also showed the same thing, that if you add glutamate to a food, you do much more than change its taste, you change its whole mouthfeel. And it's interesting to speculate that perhaps some of these compounds stimulate not only the taste receptors, but also touch receptors. It's certainly possible, but that's pure speculation. Now I want to move to the hedonic side and just say a few words about flavor hedonics. And if I make very broad generalizations, if we take the three chemical senses, the senses of flavor, for taste, innateness is much more important than learning, I think. This is just, I'm just going to say it flat out, and then you can argue with me later. For a chemist thesis, both learning and innate are important. Most chemesthetic stimuli, as far as we can tell, are innately rejected by young infants, by other species. Yet learning is plays a very, very important role in changing that to a positive for reasons that we don't understand. And finally, smell, I would say, is the reverse of taste. That is, although there are probably innate components to why we like certain smells, that would be certain sort of flavors in foods. The learned component is I think, much stronger. And I want to take one study which I find one of the most fascinating of all studies. It's an anthropological study carried out up in the Bering Straits by some colleagues who spent 10 to 15 years studying the Inuit habit of eating a traditional diet. So here is the recipe for their traditional diet. This is worth reading. Reindeer stomachs stuffed with blood, liver, fire-roasted lips and hooves, and sweet roots, all firmly sealed inside a stomach bag and buried in a bed of willow leaves for 6 months in permafrost, essentially to rot. And then this is then eaten with fermented reindeer blood, walrus fat, and seal oil. So this is making this stuff, and this is people consuming it. And there's a couple of interesting points about this photograph. First is notice that the person who's preparing the food is wearing gloves, and the reason for that is this stuff is so stinky, so awful to people that don't like it, that they really don't want other, and it's also considered not with it in the modern world, even there. And so these people often sneak away to eat this food and then have a whole set of clothes, including the gloves, that they take off when they finished eating it. So it's an amazingly potent flavor and mostly smell, and it's amazingly unpleasant to people who are unfamiliar with it, but it turns out it's incredibly pleasant to the people themselves. So these food odors permeate everything, and one of the great things about this article was that the anthropologist talking about how, and if you know anthropologists, they're particularly ones that do cross-cultural stuff, they're willing to try and do anything. This is part of their ethos, and yet they could not get this stuff down. They put it in their mouth, they would chew and chew and chew, they would retch and often throw up. So what it appears to, very clear from this, and I can't go into the details of the argument, but basically there was a time when on the Russian side, it was the Union Soviet Socialist Republic, they were trying to turn this into an area that would be more modern. They forbade people to consume these foods. They gave them modern Russian foods that were brought in. And a whole generation of people didn't have experience with this traditional diet. Then the Soviet Union collapsed. Russia became Russia, but there was no money. And so people were back trying to consume their normal diets. And what happened was people that missed this early period of exposure to it couldn't eat the traditional diets. They just couldn't do it. And so this really is the most dramatic example I know of real-world sensitive periods where it appears that you have to have exposure to this very early in life, or you won't like it. And interestingly, they also developed cravings for it. The people that did like it developed cravings for it, and that's why they went away and hid and wore gloves to eat it. Now I want to talk right at the end about flavor transmission in humans. This is work mainly done by Julie Bonella and colleagues. And following up from this real-world example, what Julie's done is look at the transmission of flavors in amniotic fluid and in milk. And so what was shown is that both just amniotic fluid, really the only evidence has been a very few of them, but in milk, many, many things are transmitted into milk. This is well known in the dairy industry. What we argue is that the amniotic fluid in mother's milk and amniotic fluid in mother's milk are both the initial flavor experiences. This is where flavor culture starts. And we would further argue, and I know that there's people on this panel that will disagree with me, we would further argue that there is a sensitive period where these flavors take on an emotional response that's different from learning things later in life. Now, we can obviously learn to eat new foods later in life. We can learn to like them. But the emotional component, I think, maybe is particularly strong for those things we're exposed to early in life, even before where the exposure is even before we're born. Here's the experimental study. Basically, there were 3 groups. One group of mothers, pregnant mothers, was fed carrot during pregnancy, carrot juice, and then during lactation, water. There was a reverse group, water during pregnancy and carrot juice during lactation, and then a control group. And basically what she showed was that either prenatally or postnatal exposure to this carrot flavor caused the infants when they were 6 or 7 months of age to show a strong response to it. So there's this learning that's going on, and basically the view would be that that this is the sort of the pathway for, at least for early development, that there's flavors in amniotic fluid, flavors in breast milk, then there's during weaning, and in childhood. And so this is the pathway where we are exposing our children to new things. And so I personally would argue that we really ought to look at this and pay attention to not just what the nutritional components of, say, formula, but also the flavor components. And that's a very emotional and tricky issue, and so I won't say anything more about that. So let me just end by talking about the sweet and salty, and this is a question that sort of came up and it actually stimulated the beginning of my career. Does early exposure to sweet cause kids to be addicted to sweet? And the bottom line is there's no evidence to support that. For salty, it's much less clear. There is some suggestive evidence that perhaps early exposure to salt may alter the later response. But definitive research in either area is difficult because there's ethical issues. And nobody's done truly experimental studies in either one. I think there are possibilities to do it, and I think that's an area of importance. So I enumerate here a few questions that I think are important. I think that studying the multisensory components are ones that we've had difficulty, and we really don't know how one sensory system interacts with another and at what level it interacts. There's some suggestion that olfactory components can actually inhibit bitter taste receptors. So there's possibility of inhibiting bitterness by smell. CNS processing of flavor. What about novel natural flavor molecules? Obviously, the development of successful strategies is what we want to talk about to alter our food habits to more healthy. And finally, this last one, which is, there will be a symposium on this, I think tomorrow, on what the sort of remarkable discovery that these taste receptors in the mouth and olfactory receptors in the nose are actually everywhere. And what are they doing? What are taste receptors in the gut and the pancreas and in the duodenum? What are they there for? What functional significance do they have? What about bitter receptors in the nose and in the lungs? What about olfactory receptors in the skin? What are they about? And it gives a much more broader view of the importance of these flavor senses in a broader area of nutrition and perhaps other areas as well. These are some of my colleagues that did some of the work that I took credit for. And Monell is celebrating its 50th anniversary, and we are looking forward to 50 more great years. So thank you very much.
Thank you, Gary. Jerry. We have time for a few questions from the audience. And if you could come to the microphone and introduce yourself, that would be wonderful. Much appreciated.
Elizabeth Jeffrey, University of Illinois. This is an area that I know nothing about, and it's intriguing. And one of the reasons it's intriguing to me is because I look at the diet of people today, which is a very unhealthy Western-style diet. And I'm wondering if there's work going on, not with just that small number of volatiles that you showed us, but work that would persuade mothers, if you feed these vegetables to your infant while you're pregnant, is it going to make a difference to the health of society?
Yes, okay. So are people from NIH listening to this? Indeed, that's the implication of what might be the case. But it would be very naive to say that we know that to be the case right now. One of the things that I've sort of struggled with in my own life has been the idea of, if these are important, and not only in utero but during feeding and nursing, why is it that there are these strict regulations about putting flavors into infant formula? And I know the response, but it's not convincing to me. So I think that that's an area that one could look at, actually, in a larger group. The in utero stuff is hard to do, and so, but I think it's important as well.
Well, I actually think we mothers are really responsible for a number of things, right? Not only do we give our children our mitochondrial DNA, and now we influence their taste and potential future health in that manner. Oh, sorry. That was just a comment, sorry. Any other questions? I won't repeat it because it was only about mothers.
Only about mothers.
Hi, Sarah Martinelli from ASU. You said you knew the response as to why they wouldn't put these tastes into formula. I'm curious, why not? Or what's their version of why not, I guess?
Well, one version is that that they think that you're trying to make the formula more tasty, and therefore would be liable to formula feed your infant, more liable to formula feed your infant if it had these flavors in it. And so you don't want to do anything to encourage mothers to formula feed, and everybody agrees on that. So the argument is that maybe this would be pushing in that direction.
Thank you. We'll have an opportunity for further discussion during our panel.